Good afternoon, everybody. It's a great pleasure to be here. As other speakers have indicated, it really is tremendous. The first time I came to present at this conference was 5 years ago, and it really is tremendous to see that the progress that's been made in terms of facilitating the development of new drugs for Duchenne, and it really has been a great privilege to be part of that process. I previously was Chief Medical Officer of Confernece, now part of BioMarin, similar to the other speakers, have to make various forward-looking statements during the course of the presentation. Just an introduction to BioMarin. I think of any company that we could have joined forces with, this probably is the best one. It is totally focused on treating rare disease, and the panels here just indicate a number of the type of patients and the type of diseases that they are dealing with. Morris there on the right, on the left-hand side, has phenylketonuria, and that's his mother, who actually works in the commercial department in BioMarin. In the middle panel, there's a boy called Giles Bueno. He has an enzyme deficiency, which causes problems in terms of both mental and physical development. And on the right-hand side, a girl, Isabel, again, with another form of enzyme deficiency, which causes growth retardation. BioMarin has 5 different drugs on the market, all for rare disease, and brings a lot of expertise to help us as we continue to pursue Drisapersen and the other compounds in our portfolio. So this is the Exon skipping program, which was acquired from Confernece. The lead compound is Drisapersen, which addresses the largest number of boys that can be treated with the Exon skipping approach. There are another 3 drugs that are currently in clinical trials, and another 3 earlier on in development. Since we are actually undergoing scrutiny by the regulatory authorities, I thought rather than talk about any data, it might be useful to reflect on some of the lessons we've learnt in trying to develop a drug for Duchenne muscular dystrophy. I think lesson number 1 is that double-blind placebo-controlled trials are both feasible and informative for a number of reasons. One is that they do provide a gold standard of evidence to convince regulators, physicians, and very importantly, payers of a treatment effect. They enroll a synchronized population with a double-blind design to limit bias, and it's important to be able to distinguish the expectation of a treatment effect when going into a clinical trial from actual drug effect. And having a placebo in there does help do that. And of course, it's also very important to be able to distinguish any type of safety signal that might go along with the disease from one that may be drug-induced. A randomization approach naturally balances both known and unknown baseline features which can influence trial outcome. And I think it's particularly important when you're examining a new mechanism of action that a placebo-controlled trial can really establish a principle of effect. The issues you've heard with drug development is the challenges both known and unknown, and particularly the unknown unknowns. And the key learning number 2, and you've heard a lot about this, is understanding natural history is absolutely critical to being able to design and interpret clinical trials. It helps understand the treatment effect on clinical endpoints and helps with the measuring and interpretation of putative biomarkers. I think one of the most insightful images I've seen over the last year or so was one presented by Lee Sweeney at the PPMD Policy Forum back at the end of 2013, because I think this really shows the impact of disease on the target tissue. Now, our drug works on muscle cells, and it's quite clear, as you can see from this sequence of images taken from boys of increasing age, that you get to a situation where an increasing amount of muscle tissue has been replaced by fat and fibrous tissue. And in that environment, there's less and less tissue available for drug effect. So it helps try and understand the likely impact of a drug that's targeting muscle on the tissue, and particularly when you're measuring an endpoint like ambulation, and that needs to be taken into account. The other aspect that this brings up is, of course, that if you're using something like muscle biopsy to be able to detect a molecular effect, such as dystrophin, as you get with more progressed disease, so the likelihood of getting homogeneous tissue gets more and more difficult as you get more and more disease in the muscle. And it's probably no coincidence that when we've been doing our studies, the best results we've had using dystrophin as a biomarker has been in studies where we've enrolled younger boys with more preserved muscle mass, and also in the studies where we've introduced a loading dose to try and get maximum amount of drug to the muscle as soon as possible. I think the other important thing is that particularly if you do have a biomarker that may have variability, is to be able to back that up with other types of biomarkers. So these can be either things measuring enzymes such as creatine kinase, which is a marker of muscle permeability, or imaging such as MR. And again, this illustrates the need, the utility of having placebo, because creatine kinase is something that also declines as muscle mass declines. So if you see a decline in CK, you may just interpret that because to be to do with disease progression. So you really need to have the placebo arm to be able to distinguish that treatment effect from just what's happening in natural history. As has been said, another really important feature about natural history is being able to understand the nature of your patient population and the influence of disease progression on your patient population. When we started, there was very little published in terms of the natural history of Duchenne as measured by ambulatory parameters such as a 6-minute walk. And so it's been very important to see the results of long-term natural history studies to understand both the effect of age, but also the baseline function. So we now know from these are studies taken in Italy that if you have a baseline 6-minute walk of less than 250 meters, after 2 years you have a 50% chance of losing ambulation. Whereas if you have over that level, then you have a less than 10% chance. And these things need to be factored in to the design of studies. So we have been running our own natural history study. First patient was recruited in July 2012, and it should complete in 2017, not only to continue to improve the understanding of natural history, but also to be able to incorporate some of the new outcome measures that are being developed, very much in collaboration with patient groups and investigators, such as the performance of the upper limb, to be able to understand the utility of imaging, and also to look at other putative biomarkers, because we believe that the more that this is understood, the quicker that the development programs can be in the future. So apart from the work we have been doing, we're continuing to work with Drisapersen, as well as the other follow-on exons that we have in development. So we have another 3 in development. So with Drisapersen, we have been initiating and restarting studies that had previously been discontinued, both in the US and in Europe and in the rest of the world. And really the emphasis here has been trying to minimize the burden to patients. We are not requiring muscle biopsies. We're using home dosing where we can, and also introducing IV dosing, intravenous dosing where appropriate. And so studies planning to start by the end of the year or early next year is studies in the non-ambulant population, in the younger boys, and also validating intravenous treatment. So Drisapersen was submitted for FDA review at the end of April. As has been mentioned, the others, this is a major consuming process to be able to do this, where days are long and nights are short, to be able to complete these submissions. And earlier on this month, we also made a submission to the European authorities. As far as the follow-on Exon programs are concerned, we have an extension study for the 44 skippable population, as well as programs ongoing for 45 and 53. As has been mentioned, the issue with a rare disease with multiple mutations is that you don't want to leave any child out. And most of the work has been focused, as might make sense, in the populations that are most common, but we understand the need to be able to address populations with much rarer mutations. And so the approach we're taking to this is actually moving away from single exon skipping to multiple exon skipping. And we have programs targeting initially the lower end of the gene, but also plans to target the hotspot as the more frequent hotspot there. So in conclusion, I think everybody agrees there's been a huge amount of progress made over the past 5 years. Drisapersen has now been submitted for regulatory review both in the US and Europe. There are extension trials underway, and also in preparation for former participants of Drisapersen programs. New studies in non-ambulance in those under 5 and for IV route of administration are in planning, and clinical studies are ongoing in the follow-on exons, and multi-skip approach for rare mutations are underway. But again, just to emphasize that this is a real collaborative process, and most of all, we're grateful to you, the patient community, to helping us to do this work. When exon skipping first was thought of, there was no funding available, and it was the patient organizations that provided that funding, because the regular grant authorities thought this was a completely fanciful approach. So thanks to you, this whole program is where it is. Thank you very much.